Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about tissue processing and embedding, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A ideas, evidence ideas, reflective statement, and some helpful tips to make candidates' portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that candidate needs to be achieved when this submodule is completed. These are relevant to each submodule. For this submodule, the expectations are that the candidate will be able to process, embed and quality control of routine histological tissue samples. Gain clear understanding of the principles and practice of tissue processing and embedding. Understand the role of processing and embedding in the investigation of key pathological conditions and how these may be investigated histologically. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. The first learning outcome is, explain the principles and practice of paraffin wax tissue processing for a range of tissues. Candidate needs to explain principles and practice of processing. Does the candidate know the principles and practice of wax processing? Do they know the steps involved in tissue processing? Do they know the reagents used in tissue processing and why? Do they process various samples the same way? Candidate could consider principles of tissue processing like fixation, dehydration, clearing, infiltration, reagents used during processing, practice of tissue processing like sample size, time, temperature, vacuum, agitation, processing schedules for fatty tissue, routine tissue, biopsies, megablocks, or rapid run. The second learning outcome is, discuss the influence of processing schedules on the quality of sample preservation for subsequent analysis and give an example from practice. Candidate needs to know the influence of processing on sample preservation and subsequent analysis. Does the candidate know how processing influences sample preservation? Do they know the effects of processing on subsequent tissue analysis? Do they process every tissue sample the same way? Do they use different processing schedules? Do they know what would happen if wrong tissue schedule was selected during tissue processing? Candidate could under underfixation and poor morphology, zonal fixation, poor staining, poor nuclear detail, autolysis, overfixation and antigen masking, nucleic acid degradation, poor staining, poor dehydration and poor wax infiltration, brittle tissue, mushy blocks, difficult sectioning, poor clearing and poor wax infiltration, mushy blocks, difficult sectioning, poor antigen retrieval, decreasing yield of high molecular weight DNA, unreliable staining results like companion markers. The third learning outcome is, discuss how to troubleshoot processing errors. Candidate needs to be able to troubleshoot processing errors. Does the candidate know what errors could occur during tissue processing? Do they know how to resolve or prevent processing errors? Do they know what effects could they have on subsequent analysis and tissue reporting? Candidate could consider the key areas of processing like poor fixation, dehydration, clearing and infiltration, different types of problems like poor staining, poor morphology, mushy tissue, brittle tissue and poor sectioning, possible measures to resolve problems like reagent change, schedule optimization, training, or test runs, steps taken during troubleshooting like problem identification, data gathering, root cause analysis, corrective and preventive actions, communication, monitoring and documentation. The fourth learning outcome is, compare the advantages and disadvantages of different processing reagents and schedules. Candidate needs to compare advantages and disadvantages of processing reagents and schedules. Does the candidate know what reagents are used during processing? Do they know their alternatives? Their advantages and disadvantages? Do they use various processing schedules? Do they know the advantages and disadvantages of different processing schedules? Candidate could consider each reagent used during processing, alternative reagent that could be used, their advantages and disadvantages, mode and speed of action, price, health safety considerations, quality considerations, compatibility, reliability, flexibility, associated problems, and various processing schedules reagents could be used for. The fifth learning outcome is, discuss the effects of heat, pressure, and vacuum on tissue processing times and tissue morphology. Candidate needs to know the effects of various factors affecting tissue processing. Does the candidate know what effects do heat, pressure, vacuum have on tissue processing times? Do they know affect the tissue morphology? Do they know the advantages and disadvantages of heat, pressure, and vacuum on tissue processing and morphology? Candidate could consider heat, pressure, and vacuum and their effects on speed of reactions, diffusion rate, penetration, time and morphology, their effects and tissue shrinkage, hardening, brittle, distortion, antigen damage, uniformity, quality of staining. The sixth learning outcome is, explain the principles and practice of resin processing. Candidate needs to explain resin processing. Does the candidate know what are the principles and practice of resin processing? Do they know the steps involved during resin processing? Do they know the reagents used during resin processing? Do they know which samples can be resin processed and why? Candidate could consider principles like fixation, dehydration, infiltration, polymerization, equipment used, practice of resin processing like sample size and type, time, morphology, antigen preservation, ultrasectioning, considerations of advantages, disadvantages, safety, and quality of resin processing. The seventh learning outcome is, explain the principles and practice of a range of tissue embedding techniques, and discuss potential errors, and their clinical impact. Candidate needs to know about various tissue embedding techniques. Does the candidate know what are the principles and practice of embedding? Do they know the potential errors associated with embedding? Do they know the clinical impact of poor embedding? Do they know how to troubleshoot poor embedding? Candidate could consider embedding techniques like paraffin wax, cryo-embedding and resin or plastic embedding, errors with orientation, air bubbles, sample mix-up, poor tissue support, presence of staples or stick tissue loss, fragmentation, impacts like delayed diagnosis and treatment, turnaround times, subsequent testing, patient distress, and others. The eighth learning outcome is, appraise the correct orientation for embedding a range of tissues, demonstrate candidate's application of this in practice. Candidate needs to correctly orientate various tissue types. Does the candidate know how to orientate tissue during embedding? Do they know what tissue types require specific orientation during embedding? Can they explain their tissue orientation technique? Candidate could consider importance of orientation, various tissues like tubular structures, epithelial tissues, hollow organs, membranous tissues, multiple biopsies or frags in a cassette, inked or bisected samples, punch or core biopsies, samples with specific special instructions for embedding like alopecia or temporal artery samples, nerve and muscle samples. The ninth learning outcome is, discuss the risks and hazards associated with the use and disposal of processing reagents and embedding media. Candidate needs to know about risks and hazards associated with processing and embedding reagents. Does the candidate know what are the health and safety risks and hazards associated with reagents used during processing and embedding? Do they know how to mitigate those risks and hazards? Do they know how to minimize health and safety risks and hazards? Do they know of any safer alternatives that could be used instead? Do they know how to dispose processing and embedding reagents and why? Candidate could consider health and safety risks like corrosive burns, respiratory hazards, toxicity, fumes, sensitization, quality risks like morphology damage, molecular alteration, equipment damage, affected subsequent analysis, disposal risks like water contamination, environmental toxicity, dangerous reactions, hazardous materials, in general COSHH, PPE, training, SOPs, waste segregation. Those are the learning outcomes candidate needs to meet for this submodule. Indicative content highlights background knowledge that might be assessed during the Viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In this submodule, candidate is expected to have the knowledge and understanding relevant to tissue processing and embedding for routine histology samples, relevant to EQA schemes for tissue processing and embedding, and be able to interpret and act upon the results. Resources section on the Brightspace contains some very useful information for this submodule. Submission of evidence section on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible question suggestions later on. Candidate also needs to select an additional piece of evidence relevant to this submodule. For evidence ideas, please have a look at the video for trainee. Each submodule also requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. These are some example questions you could give your candidate to test their knowledge, competence, and skills about this submodule. These should be based around the learning outcomes mentioned earlier. These are some more example questions you could use for this submodule. You could ask them to complete tables you created based on the learning outcomes. Or you could use images to test candidates' knowledge and competence. Supporting evidence should be selected by the candidate. It needs to be based around the learning outcomes and indicative content, and be relevant for each submodule. The evidence should allow the candidate to demonstrate their specialist knowledge and competence in each submodule. It should be specific and relevant to the candidate's in-house practice. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Each submodule also contains reflective statement, which is asking the candidate to summarize their role in the lab in the context with each submodule. How do they demonstrate that they can draw conclusion from what they have learned, reflect on their current practice and how it can change in the future due to gained knowledge? They could think about what is their lab role in context of this submodule. What impacts can their actions have on their colleagues, department as a whole, and for the patients? They also need to write a personal reflection on their training in the lab and explain why they chose their evidence. This is very personal to their own circumstance. It should demonstrate their ability to be self-aware when analyzing gaps in their knowledge. They can use it as an opportunity to reflect on aspects of their training, application of their gained knowledge and skills, and how they have achieved their goals. They should think about what their evidence demonstrates, and why they chose this specific evidence for this submodule. Also, they should write about what their training was like when it comes to this submodule and how their practice changed due to their training. Their reflective statements can also be used as a CPD evidence in case they are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, animations, pictures, posters, flowcharts, videos, photographs, PowerPoints, diagrams, graphs, infographics, charts. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like a case study, for each submodule. They could use experiments, case studies, annotated results, troubleshooting, controls production, annotated images, risk assessments, glossary, training presentation, certificates with reflective statements, nonconformances, validations, verifications, comparisons, ECHA results, annotated audits, kappa or DATIX, Q&A. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If they like experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the learning outcomes and indicative content in mind when they are selecting their evidence. Learning outcomes that require the candidate to demonstrate something would be ideal for covering with their evidence. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning requirements for this submodule. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those learning outcomes in mind when selecting questions and guiding the candidate. Bye for now.